Hello, I'm Professor Young. This is A&P II. Welcome. This is the first of our recorded lectures that you will have throughout this semester. And we start A&P II with looking at blood, uh, blood composition, function of blood, homeostatic mechanisms of blood. And so let's go ahead and kind of dive into this. Each video will be about anywhere between 15 and 30 minutes long, and you will notice that you will have embedded questions within the presentations as well. These questions do count as assignments, and you can re-watch the video multiple times, and you can answer the question sets multiple times. So it's a really good way to review for basic understanding of the content and the material that is being presented. And so let's go ahead and jump into looking at our system, our first system here in A&P II. You should remember from hopefully A&P I that blood is more than just a substance. Blood is a tissue. And so when we refer to blood, we're referring to blood as the connective tissue that it is. And it is a connective tissue because, if you remember, it is made up of cells. It's made up of ground substance, and it is made up of fibers that are actually suspended, and it's suspended within the plasma. Now, of course, the cells that we are referring to are erythrocytes and leukocytes, otherwise referred to as red blood cells and white blood cells. And you're going to learn that there's another type of cell in here that's not really a true cell. It's a cell fragment, what we refer to as platelets. And we'll get into that a little bit more as we go through the material here. The plasma, remember, is simply a watered down or a watery version of amorphous ground substance. And the fiber that is being suspended within that plasma is fibrin. And remember that it is stored as fibrinogen. And so these are all things that you should remember, hopefully, from your time in A&P I about blood as a tissue. Blood as a tissue. And then our look here is going to be to understand what makes up this tissue that we are talking about, this liquid tissue that we have. And so before we actually get into the components of this, let's talk a little bit about the functions. And the functions, I think, are relatively straightforward. We've got transportation. We have homeostatic regulation. And we have protection. When we're dealing with transportation, we are referring to the transportation of things like oxygen. We're dealing with the transportation of things like carbon dioxide, but we're also dealing with the transportations of nutrients. We're dealing with the transportation of metabolic waste. And that is including carbon dioxide, but it's also referring to things like nitrogenous waste, urea, as an example, which we'll talk more about when we get into the urinary system. But we're also talking about transportation of other materials, such as hormones. We're talking about transportation of things like um, uh, proteins. Right? And those proteins can be in the form of things like antibodies. They can be in the form of things like antigens. They can be in the form of things like albumin. They could be um, lipids, fatty acids, right? Things like cholesterol. Right? All of these things are going to be transported through blood. Heat gets transported through blood. Right? Heat is a metabolic waste. It's a byproduct of metabolism. And that heat needs to be transferred out of the body in some way. And so, very often, that heat is transferred to the blood, and then the blood carries that and it gets real close to the surface where it can dissipate that heat through our pores within the skin. When we're talking about things like homeostatic regulations, when we're talking about the regulatory processes that can go on, we're talking about things like regulating pH. pH regulation. We're talking about things like body temperature. We're talking about things like blood glucose levels. Right? And so there's a lot of regulation. Blood, just about anything levels. Blood calcium levels, blood glucose levels. These are all things that we will be talking about as we move throughout the semester. And so we see a lot of regulatory mechanisms. pH will be a mechanism of regulation that we talk about not only within blood as an introduction, but we'll get into more of that regulation in um, the digestive system. We'll get into more of that when we get into the respiratory system. And we'll get into more of this pH regulation through blood within the urinary system. And so this is a common theme that we will see as we move through the semester. And then protection. Protection is the third function that we have within this tissue known as blood. Right? What kind of protection that we have? Well, clotting is a major protective function of blood. Right? Clotting goes back to this conversion of fibrinogen into fibrin. Right? So you got a cut. If you didn't have a way to clot that cut or prevent that bleeding from continuing, you'd bleed out. You would die. And so clotting is a major function of our blood. Um, the function of white blood cells or leukocytes. Right? This is major. Right? And when we look at white blood cells, we need to also consider the role of antibodies, which we will talk more about when we get into the lymphatic and the immune systems. But altogether, our white blood cells, our antibodies, our antigens, we're talking about immune response. That is what we are really referring to here with this. And so let us go ahead and continue here our look at blood. Some of this stuff is going to be a little bit more on the basic side of things, and that is okay. All right, so when we look at the composition of blood, we see that it's got a very narrow range for its pH. And this is going to go into that whole idea of pH regulation and why pH regulation is so important within blood, because that's a very narrow range. On average, we say that the pH is 7.5. 7.4. Right? That's on average, but between 7.35 and 7.45. Remember, nothing within the body is a solid set point. Everything is a set range. Homeostasis is predicated on everything within the body happening within a range. Sometimes that range is really broad, but other times, like this, that range is really, really narrow. It only takes a couple bicarbonate ions, or it only takes a couple carbon dioxide molecules, for instance, to go ahead and shift the balance of this pH. And so this is tightly regulated for a number of reasons that will become apparent as we move throughout the semester. Um, blood varies in color based off of the oxygen content. Blood is never, ever, ever blue. That is an old wives' tale that for somehow keeps getting perpetrated within education. But understand that blood is always red. Uh, when it is more heavily oxygenated, that blood takes on a brighter red color. And when that blood is less oxygenated, that it becomes more of a deeper red color or a darker red color. But blood is always, always red. We contain about 4 to 6 liters of blood within our body. Typically, when you donate blood, you're donating about a liter of blood. And so you're only donating about 20% of your overall blood. And that's quickly replaced by the body. And the approximate temperature of our blood is about 100 degrees Fahrenheit. Now, this is an internal temperature. All right, so this is not your 98.6 degree temperature, which is the temperature of your oral cavity, right, which is lower than your internal temperature. And so that's something that very often students don't realize. You know, if you do a forehead scan, that temperature is actually lower than 98.6 degrees. And so 98.6 degrees is that oral temperature. If you were to do it under the armpit, it would be warmer. If you were to do it in the ear, it would be about the same. If you were taking temperature rectally, it would be warmer. And so the location of that temperature is definitely important. But within blood, right, because it is close to the core, and the blood spends majority of the time in the core and not at the extremities, that temperature of blood tends to be a little bit warmer. And so we, again, average about 100.4 degrees Fahrenheit. Again, there's a range of acceptability for that. And as we have talked about, or as you have started to look at in lab, we know that blood is made up of two key constituents or components. We know that we've got the plasma value, all right, which I have seen estimates and percentages anywhere from about 54% upwards to 58%. This is why you're seeing slight differences in the percentage here compared to here. It depends on the source that you're dealing with. Here, 55 and 45 is a lot nicer to deal with than dealing with maybe 58 versus 42. But just note that there will be some variability in that percentages. For exams, I don't really deal with percentages as much. Yes, you'll have to know whether or not something is normal or not, but there's a range of flexibility for that, and that'll become more apparent as we go through the material, and you kind of learn what to expect from my questions. Then we have this buffy coat, this area right there, in combination with our red blood cells. And the buffy coat is your leukocytes and your platelets. Your red blood cells are your erythrocytes. They're your red blood cells. And together they make up about 45, I'd say between 44% and 46% of your overall composition. And we refer to this area collectively the buffy coat in addition to the red blood cells as being the formed elements. We call them the formed elements because these are things that are actually produced. And they're produced within the red bone marrow. And so your red blood cells, your white blood cells, and your platelets are all produced within the red bone marrow. And so they have to be produced or formed and then released into the plasma. And this is really where the bulk of our time is going to be spent looking at and trying to understand the regulation of how do we regulate and monitor and balance the production of your erythrocytes, your leukocytes, and your platelets. You can see that within the formed elements, within the formed elements, the red blood cells are your majority element or formed particle that you're going to find. And even when you look histologically, you're going to see that. You look at a slide of a blood smear, and what you see the most of are erythrocytes. You see the most of those red blood cells. And then you will see your white blood cells, which are very evident, and you will have even fewer platelets. And so as we go through the lab material, pay attention to that. Right? As we're doing total red blood cell counts, pay attention to that. Again, all we really care about with total red blood cell counts are the red blood cells or the erythrocytes. And so white blood cells and platelets are actually lysed and destroyed when we do total red blood cell counts. Plasma, the other part of blood as a tissue. So here we go. Blood as a tissue is made up of two structures, the plasma and the formed elements. The plasma is about 92% overall water. The remaining 8% to 8.5, 9% are proteins. And so you've got some examples of proteins that are there. You've got albumin, globulins. So when we get into blood typing, we're going to talk about the ABO blood group. And you'll notice that the ABO blood group is transported, those proteins are transported on another protein that is called immunoglobulins. And so this idea of a globulin is a protein. Right? And so antibodies, transportation of iron are all done. Antibodies are a type of globulin, I should say, immunoglobulin or antibody. Transportation of iron is done on globulins. Transportation of lipids and transportation of fat-soluble lipids are usually all in conjunction with globulins. And then we have fibrinogen, right, or fibrin, which is used in clotting. That is a protein. It is a linear protein. And the majority of these proteins are all going to be synthesized within the liver, coming from hepatocytes. And that's going to become, again, that relationship between the liver, the digestive system, which is where we're getting the building blocks for a lot of these proteins, the amino acids, and the cardiovascular system will become very apparent as we move through the semester. Right. There are some other things that are usually also found within plasma. Right? And so I don't want you to think that it's only water and protein or proteins. The majority of what you're going to see within plasma is going to be water and proteins, but you're also going to have some other constituents in there. Right? You're going to have electrolytes that will be transported. You're going to have gases, carbon dioxide, things like that, that are going to be transported and disposed of through the plasma. You're going to have enzymes naturally within there. You're going to have hormones that are going to be transported. Again, within the plasma, these aren't transported within the red blood cells. These are going to be transported within the plasma. You're going to have vitamins. that are being transported, and you're going to have metabolic waste. Things like urea, things like creatinine. These are all things that will have to be moved throughout the body. And so our blood plasma, or the plasma of the blood, is the mechanism or the vehicle by which we're going to be transporting these various substances. And of course, there's going to be other things in there as well. But this is a short list of the major uh, uh, components that are also found within plasma. And this here is a good summary table for you to kind of look at and be familiar with. I do like to pull sometimes questions off of these tables for the exam, and this just gives you that better grasp of the bigger picture, right, for what exactly is happening within plasma and transportation and homeostasis or regulation of said components. And so let's go ahead and I think this is a good place to stop. And so we'll go ahead and we will stop here, kind of give you some time to digest this material, and we will come back, or when we come back for video 2, we will actually spend some time going through and starting to learn a little bit more about the specific parts of those formed elements. What is a red blood cell? What is a leukocyte or a white blood cell? What different types are there of them? How are they created and produced? What's the homeostatic mechanism or regulation of the hormonal control that is involved here? And so we'll start getting into all of that on video 2. Digest this, spend some time with it, jot down some questions. Feel free to contact me, reach out, and we'll get those questions answered up for you. And so, with that said, I will catch you guys on the flip side.